Hello, I'm Professor Sims, and in this video, I will discuss the first half of Chapter 16 on gene expression. Next week, we'll cover the second half, but this is the sixth in the series of 10 lessons held as part of my General Genetics course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing prokaryotic and eukaryotic gene regulation, so prokaryotes at the transcription level, talking about the differences between prokaryotic and eukaryotic gene expression regulation. We'll talk about activators, inducers, repressors, chromatin remodeling, histone modification, and DNA methylation, and epigenetic gene changes. Somatic cells in our bodies each carry the complete set of the genome, the genetic information in their DNA, with a few exceptions like red blood cells and some immune system cells, which lack DNA in their mature state. One intriguing aspect of this is that not all genes are expressed in all cells simultaneously, right? So this is what's leading to the disparity that we observe in a cell or in the organ functions. Despite containing identical genes, different cells exhibit different characteristics. For instance, the cells in our eyes are different from the cells in our liver or in our stomach. And the reason behind this diversity lies in the process of regulation, which maintains the efficiency of cellular functions. Expressing all genes in every cell would be an incredibly energy-consuming task, so cells wisely conserve energy by activating only the genes they need and only when they need them. This regulation also saves space within the cells, because if all the genes were expressed all at once, the cells would become massive. They'd be just like stuffed full of proteins. And by selectively expressing genes at different times, cells can respond quickly to changes in their environment or changes in what the body needs. This precise control ensures that essential proteins are synthesized promptly and precisely when they are needed, and then stopped when they're no longer required. Figure 16.1 helps demonstrate how the genetic content of somatic cells can be the same, even though the cells themselves are very different in their form and function. The differences, again, they're due to the genes being turned on or off in different cells. Imagine if this didn't happen. Like, imagine if the cells in your eyes started behaving like the cells in your stomach lining, for example. You're right, that sounds very painful. Figure 16.2 is giving an overview of all of the places along the pathway from DNA to protein where gene expression is regulated in eukaryotic cells. So in eukaryotic cells, the regulation is beginning at the very beginning in the chromatin structures in DNA, and then it continues. You have more regulation that's happening during transcription, the mRNA, and then the rate of synthesis and degradation of mRNA and proteins is also regulated out here in the cytoplasm. Now, this is true for eukaryotes, but don't forget about prokaryotes. Remember that we learned in lesson 5 that prokaryotes, they don't have the cell nucleus, so their genes are being regulated in the cytoplasm, and where transcription and translation is happening simultaneously. So there's essentially only one opportunity for prokaryotes to control how their genes are being expressed, whereas eukaryotes have this multi-step process involving transcription in the nucleus, RNA processing in the nucleus, translation in the cytoplasm. And the complexity of this process both allows for and requires several checkpoints along the way. So regulation of gene expression during transcription dictates whether certain genes are either activated or repressed to determine which RNAs are produced. After transcription, there can be further regulation during the process of translation, influencing the rate at which proteins are synthesized. Moreover, even after translation, there can be post-translational modifications that alter the structure or function of the proteins, and these modifications can affect how the proteins behave and where they localize within the cell. Figure 16.3 is again illustrating the differences between prokaryotes and eukaryotes and how they regulate their gene expression. On the left, A is showing prokaryotic transcription and translation occurring simultaneously in the cytoplasm, and regulation occurring primarily during transcription. And then side B, again, is showing eukaryotic gene expression regulation occurring during transcription, during RNA processing, during protein translation in the cytoplasm, and that there's further regulation happening through post-translational modifications of proteins as well. Table 16.1 is giving a nice little list that contrasts regulation of gene expression in prokaryotes and eukaryotes. This table in particular may be helpful to use as a resource to look back on if the stuff that we're about to talk about in the next slides starts to get confusing. Now, we're going to go into a lot more detail about these regulation processes in each type of cell, beginning with the prokaryotic cells. Within the circular chromosome of prokaryotes, there exist these blocks of genes that are called operons. Genes involved in the same pathway or function are grouped together in these operons, and they help to control regulation of gene expression in the cells. Prokaryotic gene expression is regulated by 3 different types of operons. They're called repressors, activators, and inducers. Repressors are proteins that bind to specific DNA sites near the regulated genes, and they block the gene transcription in response to stimuli. And then activators enhance transcription, whereas inducers activate or repress transcription based on the cellular needs or substrate availability. Let's take a look at the trip operon. This is an example of a repressible operon. In bacteria like E. coli, the trip operon encodes enzymes for tryptophan synthesis. When tryptophan is present in the environment, the operon is switched off since the cell doesn't need to produce any more tryptophan right now. However, when the tryptophan levels get low, the operon is activated, and this results in mRNA transcription and translation of enzyme proteins and then synthesis, ultimately, of tryptophan. The trip operon consists of 3 key regions: the coding region, so these guys here are the coding region, the operator, and the promoter. The coding region contains genes for the 5 tryptophan biosynthesis enzymes. The promoter, which is upstream of the transcriptional start site, binds RNA polymerase to initiate transcription. So this is what you're seeing here. RNA polymerase binds to the promoter, and then between the promoter and the start site lies the operator region, so that's this here. The trip operator contains DNA sequences that can bind to the trip repressor protein, which is this here. However, the repressor protein alone can't bind to the operator. In the presence of tryptophan, the 2 tryptophan molecules attach to the repressor first, and it causes the repressor to change shape so that it can bind to the operator. And this binding physically prevents RNA polymerase from attaching to the promoter and transcribing the downstream genes. So it's literally blocking the path of RNA polymerase here. Now, in the absence of tryptophan, the repressor can't bind to the operator, so the pathway is then open. In the absence of tryptophan, the repressor is not bound to tryptophan, so it's no longer able to bind to the operator because it's the wrong shape now. It can't bind. So this allows RNA polymerase to transcribe the enzyme genes, resulting in tryptophan synthesis. The repressor actively binds to the operator to keep the genes turned off, making the trip operon negatively regulated. The proteins that bind to the operator and suppress trip expression are referred to as negative regulators.
So this is what the trip operon looks like. You can see that it contains 5 structural genes: Trip E, Trip D, Trip C, Trip B, and Trip A. These genes each encode enzymes that are necessary for tryptophan synthesis, which is the process of making tryptophan in the cell. Tryptophan is an amino acid that is necessary in the bacterial cell for any protein synthesis. Because proteins are constantly being made in the cell, tryptophan needs to be present at all times for E. coli to survive. These 5 structural genes share a promoter and an operator. And then further upstream of the operon, you will find the regulatory gene TRPR. This regulatory gene encodes an inactive repressor. When this repressor is in its inactive form, it cannot bind to the operator and therefore cannot limit transcription of the structural genes in the TRP operon. In this situation, RNA polymerase can freely bind to the promoter and transcribe the structural genes, leading to the production of enzymes that help make tryptophan. In this state, the operon is said to be de-repressed, meaning the structural genes are expressed. When there's an excess of tryptophan in the environment or in the cell, it would be a waste of energy and resources for the cell to continue making tryptophan, so the transcription of the structural genes should be limited. How does this happen? It turns out that tryptophan is a co-repressor of the trip operon. It's called a co-repressor because it works with the repressor to prevent transcription of the structural genes. It will bind to the inactive repressor, changing its shape and making it active. In this state, the DNA binding region of the trip repressor is able to bind to the operator, allowing it to prevent RNA polymerase from transcribing these genes. In this way, a high concentration of tryptophan in the environment will limit transcription of these structural genes, saving the cell lots of energy. In this state, the operon is said to be repressed.
Just like in the trip operon and how it's regulated by tryptophan, certain proteins can act as positive regulators to activate genes by binding to their promoter sequences. For instance, when glucose is scarce, E. coli can utilize other sugar sources. To enable this, genes responsible for processing these sugars need to be transcribed. As glucose levels decrease, cyclic CAMP starts accumulating in the cell. CAMP serves as a signaling molecule involved in glucose and energy metabolism. And when CAMP binds to the positive regulator catabolite activator protein, or CAP, CAP then binds to the promoters of operons, controlling the processing of alternative sugars. So in these operons, CAP binds upstream of the RNA polymerase binding site in the promoter. CAP binding enhances the stability of RNA polymerase binding to the promoter and boosts transcription of the associated protein coding genes. Figure 16.5 is illustrating how the CAP protein binds to the promoter. So you have CAP floating around out here, and it, in the absence of CAMP, CAP is not binding to the promoter. But if CAMP is around, then CAP will bind to CAMP, and then they both bind to the promoter region, and it will activate this transcription. The lac operon is a classic example of an inducible operon. It contains genes responsible for acquiring and processing lactose. Lactose is a different kind of sugar, right? You have glucose we just talked about. Lactose is another sugar. There's also, galactose. Anything ending with an OSE is a sugar. So the lac operon contains genes responsible for breaking down lactose and allows cells to use lactose as their sugar instead of glucose. There's a Z gene within the operon that encodes beta-galactosidase, which is an enzyme that breaks down lactose into glucose and galactose. To activate the lac operon, two conditions must be met. First, glucose levels must be low or absent. Second, lactose must be present. Only when glucose is scarce and lactose is available will the lac operon be transcribed. In the absence of glucose, the CAP protein enhances transcription of the lac operon. When lactose is present, allolactose, a metabolite of lactose, binds to the lac repressor, preventing it from binding to the lac operator and blocking transcription. These conditions ensure that the cell conserves its energy by synthesizing lactose processing enzymes only when necessary. And it's worth noting that even in the presence of glucose and absence of lactose, the lac operon is transcribed still, but at a very low rate. Let's pause to watch a video here that helps to better illustrate how the lac operon works.
An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase, and poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy.
Now let's switch our focus from prokaryotes to eukaryotes. We'll discuss the epigenetic gene regulation that happens in eukaryotes. Now, we just went over how eukaryotic DNA is linear. It's in chromosomes located in the nucleus. Well, it's not just a long linear strand. It's actually an intricately folded macromolecule that is wound around these proteins called histones, and it forms a nucleosome complex. And the nucleosomes act like little tiny control units that determine which part of the DNA is accessible and which is not. Visually, nucleosomes look like little beads on a string with DNA wound around the histones. When we consider the organization of the entire human genome, we find an astonishing complexity. The human genome contains about 20,000 genes spread across 23 pairs of chromosomes, and each chromosome carries thousands of genes. For each gene to be expressed and its information utilized by the cell, it must be unwound and made available to RNA polymerases, which is the enzyme responsible for transcription, right? So this intricate organization ensures that only the necessary genes are accessed and expressed in each cell type at the appropriate time, allowing our cells to function in a highly specialized and efficient manner. The tight regulation of gene expression is essential for the proper functioning and development of our bodies. Transcription and translation, the two fundamental processes of gene expression, are physically separated in eukaryotic cells, and this provides multiple opportunities for regulation, as we saw earlier. The first level of regulation starts before transcription even begins, and it involves controlling access to the DNA through what is called epigenetic regulation. Epigenetic changes can modify how tightly the DNA is wound around histone proteins, influencing which regions of DNA are accessible for transcription. This early control process sets the stage for the subsequent gene expression. Transcription factors, essential proteins in this process, play a key role in regulating transcription of genetic information from DNA to RNA because they bind to specific DNA sequences and either enhance or inhibit the transcription process, depending on the cellular context and environmental cues. By influencing the activity of transcription factors, cells can finely tune gene expression so that they can respond to their needs and maintain proper function. The regulation of gene expression through these multiple levels ensures that cells can precisely control when and which genes are expressed, enabling the incredible diversity of cell types and functions that exist in our bodies. Let's watch a short video here that discusses transcription regulation in eukaryotes.
mRNA and eventually proteins are produced when and where they are needed in an organism. Some genes, such as those for housekeeping proteins in processes like glycolysis, are expressed all of the time. These genes undergo what is known as constitutive transcription. Other proteins are only produced at specific times in certain cells. In these cells, genes undergo a process called regulated transcription. Transcription is regulated by proteins called transcription factors. These factors are proteins that are produced in the cytoplasm and eventually migrate into the nucleus, where they interact with DNA and activate transcription. These transcription factors only interact with specific genes, those genes whose transcription they control. Most eukaryotic species have over 1,000 transcription factors. Regulated transcription begins when a signal is received by the cell. This signal, often a protein, begins a signal transduction cascade that lets the cell know certain proteins are now needed. The most common activation event for a protein in a signal pathway is the addition of a phosphate group. That activated protein in turn interacts with another protein. This interaction leads to the phosphorylation of the next protein in the pathway. There is often a series of proteins that are activated by phosphorylation. At the end of this cascade, the final protein will be activated and then enter the nucleus. It must make this journey because transcription only occurs inside the nucleus. After entering through a nuclear pore, the protein interacts with the specific transcription factor responsible for activating mRNA production. As with the earlier proteins, the transcription factor is modified through phosphorylation. The activated transcription factor will next bind to an enhancer region. The enhancer is a region of DNA upstream of the transcription start site that binds a transcription factor. The transcription factor then binds to the DNA and moves to interact with the rest of the transcription protein complex, located at the transcription start site. When the formation of this complex is complete, transcription of the gene will begin. It is this sequence of events that ensures genes required for a specific tissue or only at a specific time are expressed appropriately.
Epigenetic control refers to the regulation of gene expression through temporary changes to nuclear proteins and DNA without altering the actual nucleotide sequence. These modifications help determine which genes are accessible and expressed, and one of the primary mechanisms of epigenetic control is chromatin remodeling, which affects how tightly the DNA is packaged around histone proteins. DNA methylation is another common epigenetic modification. It involves adding a methyl group to certain regions of the DNA, and then this increase in methylation leads to tighter packing of the chromatin, effectively silencing the associated genes. On the other hand, acetylation and phosphorylation are epigenetic modifications that decrease chromatin packing, and these changes make the DNA more loosely wound around the histone, so they're more accessible to transcription factors and RNA polymerase, and that promotes the expression and activates the genes. Figure 16.7 is giving both an illustration and an electrograph of DNA wrapped around histone proteins to create nucleosome complexes. These nucleosomes control the access of proteins to the DNA, and as you can see here on the right, when viewed through an electron microscope, the nucleosomes look like little beads on a string. When nucleosomes are spaced closely together, they prevent transcription factors from binding to the DNA, and this effectively turns off gene expression. On the other hand, when nucleosomes are spaced further apart, the DNA is exposed, making it accessible to transcription factors. And this allows the process of gene expression to take place, as the transcription factors can now bind to the exposed DNA and initiate the transcription of genetic information. So the positioning of nucleosomes is a key determinant in regulating gene expression, ensuring that genes are activated or repressed as needed by the cell. Let's watch a clip of how the histone modifications happen within eukaryotic DNA expression.
Today we understand epigenetics as the study of mechanisms that cause changes in gene expression, but that are not changes in the DNA sequence. These mechanisms include DNA methylation, histone modification, activity of non-coding RNAs such as microRNAs, and the effect of non-coding repeating regions in the DNA code. Before we go into the specifics of epigenetic variation or regulation, let's discuss the structure of DNA and how it is organized in eukaryotic cells. As you remember, the foundational understanding about phenotypes is that DNA codes for genes. Those genes are transcribed into RNA, which are then translated into proteins. However, these processes appear to be regulated by a variety of mechanisms that are not directly recorded in the actual code of DNA. In fact, individuals and members of different species can have similar DNA, like twins, or many conserved genes, like humans and monkeys. But significant phenotypic variation can be easily observed. That is the result of epigenetic changes that are either established and passed through generations or acquired and lost throughout the organism's lifetime. The eukaryotic cell has a nucleus that contains tightly packaged DNA folded into structures that we all know as chromosomes. These are packaged together by the way of chromatin, protein complexes that the DNA is wrapped around. On these images, you can see how the DNA string has small bumps or beads. These visible beads are called histones. When these beads were originally discovered, scientists first confused them with genes, until eventually it was proven that the string-like structures actually contain the genetic code. Histones can be either grouped together, forming lumps of DNA, or as we can see in the picture on the right, relaxed and spread out. The changes in the way DNA is packaged is used for cell division when whole chromosomes need to be moved around, or to regulate what code is more or less accessible for transcription. The regulation of DNA compactness is regulated in several ways. One of these is DNA methylation. Cytosine, which is one of the amino acids, can be modified by a methyl group, CH3, attached by a specialized protein called DNA methyltransferase. In the first minutes of life, when we are composed of a single cell, this epigenetic information has been wiped clean. In the fertilized egg, the methyl groups have been removed, and every gene is like all the others. Through the process of cell differentiation, DNA methyltransferases add methyl groups to genes, shutting off some and activating others. When a cell divides, the epigenetic information must be transmitted to each of the new cells. A different DNA methyltransferase, DNMT1, adds the proper methyl groups to these DNA strands as it is replicated. The information is propagated in a tricky way. Methyl groups are almost always added to cytosine bases with these sequences CG and GC. Notice that both strands have a cytosine, so in a methylated region of DNA, both strands will have a methyl group. When the DNA is replicated, each of the new DNA double helices will have one old strand complete with methyl groups, and one strand which is not methylated. So DNMT1 just needs to look for CG base steps where only one strand has a methyl group. The basic repeating unit of chromatin is the nucleosome, in which 146 base pairs of DNA wraps around an octamer of core histones, consisting of pairs of H3, H4, H2A, and H2B. N-terminal tails of histones protrude out of the nucleosome and are subject to a variety of post-translational modifications such as acetylation, phosphorylation, ubiquitination, and lysine and arginine methylation. Acetylation was the first of these modifications to be linked with active transcription, and subsequently phosphorylation of histone H3 was found to cooperate with acetylation in translational activation. Some histone methylation events have also been associated with transcription activation and others with gene silencing. One important aspect of histones is that they can be changed to alter how much packing the DNA is capable of. There are several modifications that affect how well DNA is packaged. The basic regulation is done via groups of atoms that are at the ends of histones. These can be of several types and will have a positive or a negative charge that either attract them together or force them apart. The DNA region that is wrapped around the histones can be more or less accessible, causing variation in gene expression. Histone proteins, or nucleosomes, undergo a host of different post-translational modifications, including phosphorylation, acetylation, and methylation, which have profound effects on the remodeling of chromatin. Histone modifications can function either individually or combinatorially to govern such processes as transcription, replication, DNA repair, and apoptosis. Methyl groups of atoms increase packing, and acetyl group decreases packing. Also, phosphoryl group can be attached to the histones and cause a decrease in packing. These groups of atoms are attached to various types of histones at various locations. As the DNA is wrapped around the histones and can be more or less accessible, various positions of the DNA are affected. These include the promoter region, transcription start sites, and the introns and exons of genes. Each one of these elements can have a significant effect on the level of gene expression, on alternative splicing, as well as other downstream regulation of pathways.
Figure 16.8 is again showing how nucleosome spacing can allow or prevent transcription factors from binding to the DNA, and how histone modifications can cause a gene to be expressed or repressed. Chemical modifications of histones influence the spacing of nucleosomes, which in turn affects whether a gene is turned on or off, right? So specific chemical tags, such as phosphate, methyl, and acetyl groups, are added to histones in DNA. These tags act as signals telling the histones whether a particular chromosomal region should be open or closed. Importantly, these modifications are not permanent and can be dynamically added or removed as needed by the cell. When certain regions of the DNA are unwound and opened up due to these modifications, it allows transcription factors to access the promoters and other upstream regions of the gene. This enables the initiation of transcription, process of converting the genetic information from DNA to RNA. The presence or absence of these chemical modifications on histones and DNA can fine-tune gene expression, ensuring that specific genes are turned on or off in response to various signals and cellular needs. Figure 16.9 again is illustrating the histone proteins and how they and the DNA help control gene expression in eukaryotes through modifications. The signals on the histones and DNA determine whether a chromosomal region is open or closed, and the chemical groups like phosphate, methyl, and acetyl can be added to histone tails, affecting how tightly DNA is wound around them. And then by altering histone charge, such modifications can relax DNA binding and influence chromatin accessibility for transcription. And I am putting one more video in here, but it's very short, and it's a really cool animation of this, how it actually looks in the cells.
Our genetic code, or DNA, is directed, controlled, and regulated by epigenetic factors which surround and scaffold the DNA in order to orchestrate the cellular mechanisms of life. The epigenome is the collection of DNA, RNA, proteins, and their chemical modifications that alter gene expression without changing genetic code. Discovery research in epigenetics seeks to understand how the environment influences regulation of genes packed within our chromatin, resulting in diseases such as cancer. Epigenetic machinery modifies accessibility of regions in DNA by adding marks to the tails of histones to create epigenetic patterns of chromatin modifications. The addition of an acetyl group to a histone tail changes the electrochemical charge of the histone, causing it to relax and release the DNA. This makes the DNA in that region more accessible to transcription machinery and allows that gene to be expressed. Placing a methyl group on histone tails can either increase or decrease that pattern of gene expression, depending on where the mark is placed. Additionally, placing a methyl group directly onto the DNA results in permanent shutdown of that genetic region. The epigenetic modification patterns of chromatin change during development and differentiation and can respond to environmental changes. When cells are exposed to environmental stimuli, the patterns of chromatin modification can become unregulated as cancer cells hijack epigenetic molecular machinery. More than half of human cancers contain mutations in DNA-associated proteins that are involved in chromatin organization and regulation.
Now, we're going to discuss in the next lesson many more types of gene regulation that occurs in eukaryotic cells. But for now, thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.